Hello everyone, I'm Mike Ward and welcome to Conversations in Healthcare, a series brought to you by DRG, part of Clarivate. In this series, we have an opportunity to hear the challenges and opportunities that are facing the healthcare industry, particularly from business leaders who are at the cutting edge, at the coalface. And it's really interesting to hear what they've got to say, particularly when events are sort of moving very, very quickly all the time. So in line with this, I'm delighted to be joined by Caroline Loew, who is the president and CEO of a company called Glympse Bio. It's a private biotech based in Cambridge, Massachusetts, which originally sort of spun out in 2015 from MIT. And the company is focusing on the better understanding of disease to transform both the detection and predict sort of treatment responses using bioengineered sensors that have been designed for each protease mediated disease. Earlier this year, the company closed an oversubscribed Series B financing at $46.7 million. And that is going to be used to support the development of the company's biosensor platform in fibrotic diseases such as NASH, or non-alcoholic steatohepatitis. You can see why everybody calls it NASH. As well as explore the potential for use in oncology and in infectious diseases. So, Caroline, I hope you and those you care about are keeping safe and well, and thanks so much for joining us.
Thank you. Real pleasure to be here today. Thank you for the invitation, Mike. Really appreciate it.
So, to kick off the conversation, I thought actually it'd be really great if you could sort of just explain the platform that actually underpins Glympse Bio, how it's designed to provide that better predictability of treatment responses.
Right, great question. So as you said, we have a diagnostic that is designed to both better detect disease as well as determine response to treatment, and we see very broad use for that. Right? If you think about you may have experienced this personally or with a family member, just this whole issue of how to diagnose disease and how to find out whether you have the right treatment and the treatment is working is just really an underserved need today. And so our technology is protease mediated. So there are over 570 proteases in the body, and they're involved across all aspects of human health and disease. And we've developed a technology, it's platform-based, that identifies which proteases are involved in a specific disease state. And then we have sensors that we send into the body that are tuned to the specific disease state. They can detect the disease state, the disease activity specifically. So they're sending out real-time signals on the disease status. We then collect a urine sample an hour after the administration of the sensors, and we're able to tell first of all what the situation is with the disease, the status of the disease, the progression of the disease, and then also we're able to determine whether what the response to treatment is. As you mentioned, our first indication is in NASH. And we focus on NASH because it was a disease that has a significant unmet need. There are a lot of therapeutics under development. There isn't any treatment today, but there's also no good way to detect the disease either. So we sort of have this dual problem. Today, the main way to detect disease is actually with a liver biopsy, which is incredibly painful. And is not something that can be repeated. And so to have a simple kind of non-invasive test that can both detect the disease early, and then track the treatment response is something that we believe will be particularly valuable.
And so NASH is the first sort of indication. Is it because the liver biopsy is so intrusive and unpleasant that you decided that would be a sort of almost like a good poster child for for the platform?
Absolutely, a couple of different reasons. So that's one good reason, right? You know, high unmet need, you know, real need for a diagnostic. The current diagnostic, the liver biopsy, is actually not particularly accurate either. So there's also a need for a good diagnostic. It's also something that it can't be used serially. And so you're not able to track the disease over time. So that's the second reason. The third reason is that NASH itself is a highly protease-mediated disease. And so as a target for our technology, it also made good sense scientifically. And generally from a portfolio perspective, these are the kinds of things that you would look for. You would look for, you know, high need in terms of, you know, high medical need in terms of patient care, but also good overlap for your portfolio as well. And in terms of protease-mediated diseases, while there are many, that's one where there's significant need. Another, you know, potential interesting area is oncology. The fundamental pathway of tumor killing in oncology is actually protease-mediated. So it's another good example where we can potentially deploy our technology. And so we follow the science, and you know, protease mediation, as I said, is quite ubiquitous in disease. And so it's sort of a question of where's that overlap, right? Where's that need to really diagnose disease, as well as, you know, good overlap with the protease mediation.
Right. So yeah, one of the things is that yeah, last year, it was about this time, sort of in the fall of 2019, you entered into a strategic collaboration with Gilead, Gilead Sciences, in this NASH space. So you know, listening to you know, what you said, could you sort of explain, you know, I guess first how that deal sort of, you know, came about? You know, how was it that, you know, Gilead was the partner, and also how it fits in with that, you know, that deal fit in with your business model. So, you know, what you're doing and how you're going to benefit, and what Gilead's doing and how they benefit.
Yeah. So we actually had, we have been working with Gilead for quite some time, predating that deal. We had an undisclosed partnership with them prior to that, which was in the preclinical space. And so they had actually been tracking our technology for quite some time. And this is sort of generally how we see the evolution of our technology with pharma companies. So my background is actually in drug development. I'm not a diagnostic developer, but sort of how I came to Glympse was really trying to solve a problem that I had seen on the therapeutic side, which is we develop all these great therapeutics, but one of the big barriers to therapeutic development is being able to both identify the right patient and also to track therapeutic response. You know, one of the, so particularly if you look in early pipelines, it's a really, really big issue. Generally, we're saying, you know, fantastic science, but essentially our ability to decode that biology is being sort of held up by the absence of really good diagnostics. And there's been a lot of research applied at that over particularly the past 5 to 7 years, but you know, it maybe hasn't advanced as much as it could. And I was very frustrated with it in the role that I had in my last company, Bristol Myers Squibb. And so the idea to come to a company like Glympse to maybe really advance that frontier, to find diagnostic tools that could have broad applicability in the end for patients, but before that in the drug development arena, was really quite compelling for me. So back then into the Gilead question, Gilead is a leader in NASH therapeutics. They had been at the forefront of that for many years, and so they were a natural partner for us. We have been in conversation with them, certainly predating my time as CEO, I think since about 2015. We had a preclinical collaboration with them. The results of that were actually published at a major liver meeting last year. And so they had sort of been tracking our technology. We then had a collaboration that was preclinical, and then the results of that were so compelling to them, they then moved into a clinical collaboration. So it was an iteration, and those kinds of iterations are very common in sort of the biotech ecosystem, right? Companies, large pharma companies or biotech companies will build collaborations with small companies. They'll watch the evolution of the technology, and then as the technology matures, they will enter into large clinical collaborations. So this was very natural. And for us, we wanted to be with a leader in NASH. We wanted a leader who had a broad portfolio in NASH so that we could really look at the true application of our technology. And also it enables us to learn, right? We're a small company, we don't know everything. And there's a lot of advantage for us to work with a large therapeutic leader. So sort of the duality of the technology is really the following. We are looking to do two things with our technology. The first is just to be able to identify patients, right, who have disease. And the second is then to understand therapeutic response. We can identify patients who have disease potentially by ourselves. We can't undertake therapeutic response without collaborations with pharma companies, right? So a collaboration with a company like Gilead is perfect for us. But the other piece is that pharma companies have a problem to solve. They can't medicalize the market. They can't introduce therapeutics meaningfully into the market without a non-invasive diagnostic that helps them identify treatable patients and determine whether they're responding to treatment. It's the classic way the osteoporosis market opened up, for example. It was with the presence of the DEXA scan. And this is sort of the advantage of the collaboration. And so we have other collaborations and discussion. Companies are really, really interested in a technology like ours because it's going to enable their therapeutic to meaningfully come to patients. Without being able to identify the patient and determine the treatment response, there's no market. So there's a real symbiosis. And I've learned from all my 20 plus years in pharma that collaborations are born out of symbiosis, right? Scientific symbiosis, but also in the end that both companies create some value together. In this case, we're sort of the opportunity in the marketplace.
So the deal. I mean, it sounds like the platform. It can be useful. In NASH, is the way that you're sort of structuring the deals that a partner would have exclusive use of the technology of the platform in a particular indication?
Generally not. I mean, if those are business terms in the end, but if a company came to us and was interested in that, of course, those are business terms that we discuss in any collaboration. But in a situation like NASH, I think the way that many companies are approaching discussions like us are sort of like the notion of a rising tide lifts all boats. The absence of diagnosis and treatment response in the marketplace is going to be a real barrier to medicalization, period. It's not a problem for just Pfizer or just Bristol Myers Squibb or just Gilead or just Novartis. It's a problem for everyone. And so unless everyone contributes to solving that problem, the market isn't going to open up. And today, just in the US alone, it's estimated between 16 and 20 million patients with NASH who are undiagnosed. Globally, that's estimated to be 100 million. Those are patients who are progressing predominantly slowly along the cirrhotic spectrum. So sometime across the NASH spectrum towards cirrhosis. So that's like a 10 to 20-year journey. There's a subset who are progressing extremely quickly. And when those patients generally get diagnosed, they're cirrhotic, right? They need a liver transplant. That's a terrible, terrible diagnosis to have. And some of those patients can't be, are untreatable, unfortunately. And it's a terrible, insidious disease. It's called the silent killer. So figuring this out, I think, is a problem that the scientific community is trying to own collectively. You see the hepatologists trying to own it with the with the pharma companies. There's a lot of momentum around it, and the diagnostic is one pillar of that. The diagnostics is therapeutic. There's also the notion of building disease knowledge as well.
So you mentioned, I mentioned in the introduction, and you followed it up that sort of NASH is the first area, but in fact you're looking at sort of opportunities in other therapeutic areas. How are you going to decide, or how are you deciding which ones you're going to target first?
Yeah, it's sort of the perennial it's the perennial portfolio question that every company faces, right?
Yeah.
And I can tell you, so my discipline, right, my background in particular has come from managing large portfolios in pharma companies, and so I've spent a lot of time addressing these questions and thinking about these questions. And it's never binary, right? It's never sort of, sort of a simple set of like yes, no kind of answers. So you're looking at a complex set of factors that you're trying to weigh together. But fundamentally, you're always driven by the science, right? You're always driven by where your science or your technology really has the ability to differentiate. And how we think about it for us is sort of like what we call proximal diseases. And so in the fibrotic space, or the protease mediated fibrotic diseases, and there's sort of a cluster then of related of protease activity. If you think about NASH as a core, there are a cluster of diseases that have proximity, and pharma companies sort of follow this in their portfolios as well. So you sort of have NASH, you have oncology, you have immunology, you have infectious disease. And if I tell you that that's kind of a cluster, there's protease mediation in all those diseases. And if you went back after our conversation and you said, well, that's interesting. If I looked at the pharma companies and I said, are most pharma companies who are in NASH also in those diseases, you would find out the answer is yes. We're probably in 3 of those 4 diseases. And the reason is that the fundamental biology is very similar. So there's a lot of like proximal nature to the biology. So we're following that premise too. We're focusing on oncology. We're also looking at infectious disease. We have very, very strong data in infectious disease. And so we sort of see the same basis for us. I think what's interesting for us is because, we know that proteases are so broadly involved in human health and disease. We see at some point kind of the potential to have, broad screening ability and to be able to look, very broadly at protease enablement or protease engagement in many diseases, and potentially diseases where there's no good diagnostic today. But I say we're cautious. We maintain focus, and I think always the risk, any company, but particularly for a small company, is I call it spreading the peanut butter, right? You spread the peanut butter too thin. You end up you get a tiny little bit done of a lot of things, and you don't achieve anything. So we're very focused. We started our oncology program. Shortly after we had very good data, and we're clearly going to the clinic with our NASH program. We've started infectious disease post our Series B. So we're being very measured in making sure that we have the ability to really, first of all, fund how we move forward, but also, gather good data and progress the program in a meaningful way. So it's a very thoughtful process, I would say, but it's driven fundamentally by the science. And in the end, the need to build a diagnostic in the space. And I would say infectious disease, just as an example, you only need to look outside right now to know that, diagnostics in infectious disease are an important opportunity.
Yeah, sure. So looking at in sort of the background when I was doing my homework for this interview, I noticed that your technology, it combines synthetic biomarkers with machine learning to sort of help identify and monitor the progress of these diseases. What have been the challenges, sort of in validating the approach? And I mean, there's always a bit of sort of skepticism around, artificial intelligence and machine learning. So, how have you sort of, dealt with that kind of skepticism as well?
Yeah, it's a good question. I think there's a really important distinction here, and I would be the first to draw it. This universe of kind of like machine learning and artificial intelligence is an enormous spectrum, and those terms are used, I think, with abandon. We have a machine learning classifier that we apply to our data, and we teach it, and then analyzes our data prospectively, so it makes a prediction on our data. It's very rudimentary. I mean, in the context of machine learning, it's absolutely validated, it's absolutely predictive, and of course, we build it and we validate it and so forth. But in the context of machine learning and artificial intelligence, is very rudimentary. We don't have enough data to make it more sophisticated than that. We will when we go into large patient cohorts, and eventually when we go into the total patient population, we'll be able to make it more sophisticated, and that's our expectation. But today it's not. It's pretty rudimentary, and it's a kind of thing that's used pretty broadly. For us, we have been, and one of the reasons that we didn't talk broadly about our technology sort of post our Series A until our Series B, was because we wanted to make sure that we had built a very broad cohort of preclinical data and had human clinical validation of our technology. So essentially, at that interface that you're talking about, sort of biology, machine learning, and bioengineering, that we had demonstrated clearly, and we had demonstrated across multiple animal models and into humans that this technology really worked. And we have that, and in fact, we're publishing our human data at the upcoming liver meeting in a couple of weeks. But we had that sort of complete package by the time we went to our funding at our last round. And that essentially was the validation, right? So we took all the risk off the table, which was to say, we've now traveled through multiple different animal models. We've used this combination, we've gone to humans. And so what's left now is just simply efficacy risk, right? And so does it really work? And that's a classic therapeutic risk, right? That's part of development, that's part of drug development. So yes, of course there's skepticism, but we're not doing anything that's wildly complicated. And we went through extensive validation, and I think that's sort of the key. Again, I came from a drug development background. I'd seen more failures than I care to talk about, and those, those are valuable learning lessons, right? You learn more from your failures than you do from your successes. We had some wonderful successes, but I don't really count those. It's the things that you learn from the things that don't work that really teach you, and I brought those lessons to this, and it's that led us to build, I think, a very robust data set with multiple sets of validation across multiple different models, and then finally the human model, and then we were able to say, okay, we have confidence that this works.
So actually, you talked about your background. You joined the company as president and CEO in November 2018. Yeah, having spent the previous 11 years at senior roles, most recently at Bristol Myers Squibb, but before that at MSD. What was the biggest challenge from going from that big pharma environment into, what at that time was, I guess, a 3-year-old, sort of spin out?
Yeah. I had really thought a lot about this. I had, I think it's actually maybe a lot of women are, and I had been really deliberate in my career trajectory. I'd always wanted to run a biotech. And I had sort of had this very deliberate career plan where I felt that there were a set of experiences that I needed to build. And I had kind of moved from role to role, kind of gathering those experiences. And my role at Bristol Myers Squibb, where I sort of had the number 2 position in the R&D organization, sort of running the whole of the R&D strategy and operations and portfolio. And I felt to me it was kind of like the capstone, which was like, I'm going to see the totality of an R&D operating organization, and when I've done that, I feel like I can go and sort of run a biotech. And so it was sort of, that was the last step for me before I left. And I spent about a year before I left. I was planning to leave, and I spent about a year before I left. I had very fortunate, I had a broad network. I talked to lots of other CEOs. I looked at sort of the spectrum of different types of companies that were public, companies that were still private, companies that were very early stage, like the company I went to. companies that were at later stage, trying to figure out what was right for me. And, you know, in the end, at my heart, I'm a builder. I like white sheets of paper. And I decided that a very early stage company, so when I joined Glympse, I think we had 4 employees. We had essentially shown that the scientific concept of the technology worked, and that there was a sort of a business model there. I had just raised the Series A round, and I took over then. And so the whole premise for me was then building the company, which was what I really wanted to do. And I think, you know, when you think about it, if people are contemplating this kind of move, it's really about understanding, first of all, what your strengths are and what you want to do. And then secondly, finding the company that kind of meets those. So for me, it was very, it was very deliberate. It wasn't, you know, a kind of a sort of knee jerk, but there was, you know, it was a process that took a long time with a lot of consideration, and the technology played a lot into that. It was solving a problem that I had been very frustrated with when, and grappled with a lot in particularly my last role at BMS.
So that's kind of interesting, because you know, so I was sort of thinking, so what advice would you give, you know, other pharma execs who, you know, think of making a sort of, you know, a similar career move? But it seems like there was this huge amount of due diligence, and it was almost like an entrepreneur. You spot that there's a problem in the marketplace, and then you try and find a solution for it. Are there other things that, for example, were there sort of key skill sets that, you know, you acquired, maybe unintentionally acquired, that actually have been very, very useful, you know, as you sort of transitioned and you know, started to build this biotech?
You know, it was interesting. I had a conversation with a colleague about a year ago, actually, and one of the things that I observed, there's a huge outflow, right, from big pharma into biotech, and I think a general assumption that people can make the transition, which I actually don't think is true. And so I think sort of what's behind your question is that people do have to understand that in many cases it's different. That it is much less structured. That you have to be, you're going because it's by nature, you know, it's a riskier endeavor, that you really need to, you have to want that agility, right? The adaptability, the you know, the lack of structure and the fluidity. I thrive on that. I thrive on change. I mean, I don't change for change's sake, but I like the fact that we can make decisions and then we can act on them right away, right? There's not an enormous bureaucracy. And certainly some of my peers from BMS in particular who've moved have also thrived on that, right? They, you know, BMS was actually a pretty agile organization as well, but it's still markedly different. And so the idea that you can have that, you know, in a less structured environment, that you can really drive the opportunity and the scientific opportunity when you see it is really great. But for some people, that's not right. And I think that's actually very important. So you have to be true to yourself, right? And that's the key. Like, so same with any job, you know, understanding, you know, what's important for you and what makes you tick is really critical. I mean, I think there's an allure, there's no question. Everyone wants to be there. But you should be honest, right? And it might not be the place for you, which is fine. We need the ecosystem, and we need equally talented, you know, scientists and physicians, you know, in large pharma as well, and large biotechs. So there's room for everyone.
Yeah, I mean, I guess again, as a sort of as a leader within a pharmaceutical company, there are always going to be times when you are having to push for more resource to sort of, you know, move a program forward, etc. But you know, you mentioned that you joined after the Series A, and you know, the company has just done a sort of Series B financing. And I just wonder whether, you know, was the sort of the questioning that you were getting from, you know, investors and potential investors, much different from what you might have been receiving internally within sort of pharmaceutical companies? And, you know, and what are the hardest questions normally you have to answer?
Yeah, I would say the rigor of training that I had coming from a large company actually helped tremendously. I mean, it conditions you in a way that I think is pretty unique, particularly kind of the type of role that I had, you know, to really understand your financials, to really understand your data, to really present it in a very pithy, focused way. So I think all of that was a big plus. It meant that we went into our Series B with, I certainly felt very confident, like a very comprehensive set of data, B, a very well structured set of data. And I felt that we were pretty well positioned to answer any questions. I mean, a couple of interesting things I think happened to us. You know, we were looking for a particular type of investor in our Series B. We wanted people who were really going to be able to contribute to the growth of the business. And I'm sort of always of the mind that you want people who are going to challenge you. And so we actually liked it when we found investors who really, you know, who really pushed us and maybe asked questions that we hadn't thought of, or, you know, looked at our data a little bit differently and caused us to go back and reanalyze it. We didn't find anything that was particularly, I don't recall anything that we didn't know, but we certainly on a couple of occasions had to go back and reanalyze data for investors. And for me, that's really interesting, right? When they look at something just, you know, in a different, tangentially to the way that you have, I find that very interesting. And we certainly found it very interesting. One of those investors ended up leading our round. So I think that tells you how much we welcome that. Instead of shying away from it, we actually embraced it, and we have very good dialogue with them. So overall, it was a very, you know, it was a very positive thing. But for us, the more sort of interesting experience, I mean, interesting experience was that we signed our term sheet sort of right after the pandemic hit, literally like days after lockdown. And so we had assembled kind of a group of investors who we knew were going to follow our lead investor. And I don't know if you remember these, it's hard now actually to remember that this happened, but you know, in the US, and I believe everywhere, the stock markets crashed, and then there was a massive crash. And so we signed our term sheet actually the day the stock market bottomed. So it was the lowest day. And we had investors who were planning to come into our round who had massive capital markets exposure. And also then other investors who were just, their companies were, you know, kind of, you know, in panic, and, you know, who knew what was going to happen. You know, they were worried about protecting their existing portfolios. It was this period of probably like 2 months of just complete, I won't say mania, but it was very panicked environment on the investor side. And so we had a number of investors who backed out with good reason, right? They were either, their capital markets exposure was massive, they lost a lot of money in the near term. They had huge issues with their portfolio companies, and they decided to reallocate to their portfolio companies. And so for us, then rebuilding very quickly a syndicate. So you saw we end up so oversubscribed, it worked out fine for us. But rebuilding a syndicate in a really short time in that environment where everyone was very, essentially, I mean, scared is maybe not too, is the wrong word, but everyone was very nervous and no one knew what was going to happen. And our whole investment pitch had to change. And that was, I think for us, was interesting. It went from being the promise of the technology and blah, 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 to if this environment persists, like where we are right now, this dire financial situation, and you have to dramatically change your plans to make your investment last from like X years to Y years, or, you know, something happened, you know, it was every apocalyptic downside scenario you can imagine. That was all they were interested in. And so, you know, for us, like getting in their mindset and saying, okay, we're going to actually proactively go out and address all of these questions was very interesting. And so for me, that was the, I mean, I guess it was interesting. It was very, I mean, it was eye-opening, to put it mildly. But I think how we got through that and how we ended up where we were was because we stood in their shoes, and we said, instead of saying, which I think would have been easy, which is like, oh my God, like the sky's falling and we're not going to get the money. We immediately pivoted and we said, we understand what's happening and we're going to proactively go out and solve their problem for them. And that sounds a bit glib, but to say, we're going to understand how they want to perceive us. And if we can make that, if we can give them confidence that we as a management team can manage any scenario, then the underlying value of the technology, which had led us to build a really good investor syndicate before, should still be there. And that was what got us through. And that was what ended up with the investment round being what it was. So that was very interesting. And it's interesting for me. I look now at how buoyant the markets are, and how many rounds are going through. It's really easy to forget that there was this moment where nothing was happening. There was sort of this paralysis. That was an interesting experience. Yeah.
Yeah, it was just sort of a 2 month, 3 month sort of
right.
Area where everything sort of seemed to quiet down. But clearly you got your message across because the subsequent round was oversubscribed.
Exactly.
So so although people obviously were concerned about the uncertainty, maybe the way that you had pivoted worked.
Yeah.
So you joined a couple of years ago. And you described earlier on about how you're a builder, so you're looking to sort of build a business. How has the business changed in the past couple of years, and now with this big tranche of finance, what sort of milestones can we anticipate Glympse achieving in the foreseeable future?
Yeah, it's been, the past couple of years have been exciting. So, we grew a lot as a company. We became a clinical stage company. So we substantially validated our technology in NASH and achieved our first in human, and the data is about to be published. So that was very significant for us. Sort of a broad set of preclinical data. We demonstrated that we can detect disease better than any other technology across the fibrotic spectrum. We can determine drug response, and we validated that, and we've achieved our first in human. So all of that was great. We also advanced our second indication, so we have some great early validation in oncology. So that was sort of the Series A window, and then for Series B, as we look forward now. The really big thing will be, we expect by the end of our Series B window to actually have an approved indication in NASH. And so that's very exciting, right? Well, there'll actually be a commercial stage company by the end of that window. So that's the big sort of next step. We also expect to have very substantively advanced oncology program in clinical development as well. So everything continues to step forward, but the really big step over the Series B window is the NASH program getting to hopefully an approved indication.
All right. So this has been absolutely fascinating. So as a sort of final question, I mean, and kind of alluded to it, that 2020 has been a sort of extraordinary year. The way that people and companies have had to adapt to the various changes. So I was just wondering, what are you now doing differently as a CEO? That behaviors that you may have introduced recently that may become sort of permanent and actually are going to be important as you manage the business going forward.
Mm-hmm. Yeah, it's a good question. At the start of the pandemic, we were fortunate that very early on, in like late December, early January, we started to see what was happening in China because we were manufacturing our clinical supplies 200 miles from Wuhan. So we saw what was happening, and first of all, we were dealing with that issue in China, but we also started to plan pandemic response for our US operations. And it was largely because I had actually had responsibility for emergency preparedness at Bristol Myers Squibb. And so he'd kind of learned this. I had this muscle, if you want. And it sort of, it kicked into gear immediately. It was hard for me to believe that we could see what was going on in China and that this wasn't going to be a bigger issue. So we sort of kicked in, 3 things that I think, everyone sort of has in their mind, but we've now kind of ingrained in the company in a way that are, I think extremely prominent. The first is, how carefully we take care of our team and how responsive we are to their situation. So we are, I think, likely to be in what's a permanent optional remote working environment. So we have a lab-based organization. We have offices. We give people the option to go in, but it's an extremely volatile situation. We have people who have kids, who have partial remote schooling or remote schooling and all sorts of complexities. So we've said people should work however it makes sense for them. We don't have any mandate on how people work. Do what accommodates you. And what's great is the whole company is rallied around this idea that people, we have very, the same as every company now, probably very good collaborative working structures remotely. And so we're making sure that we really adapt the organization's operating model to institutionalize that idea. So that's one thing. And we're very kind of focused on the organization and making sure that we really support our organization in that, and that's of course beyond all of the regular just health and safety type things. So that's one thing. The second is to make sure that we focus on, we prioritize ruthlessly. And I think it's what we learned early on was that, particularly when we stopped working for a short period, when everything was shut down, that was about 2 weeks, that you have to be really rigorous, ruthless, and to stop work that isn't important and to redirect people's priorities. But that has led us to kind of pull through much more rigorously how we prioritize and direct the organization. I think as a biotech, you already have that. But it was something that we sort of engendered, I think a lot more deeply in the organization. So, a lot more around priorities, and then finally planning. Just as we look forward, we became a lot more flexible around our planning, and a lot more contingency planning was introduced as well, just because you don't exactly know what's around the corner. And so we just become a lot more mindful, I think, about those 3 areas. And it certainly served us well. We haven't missed a single milestone since the pandemic started. And, the work, our team has certainly responded fabulously to, what's been an extremely difficult situation.
Right. So look, Caroline, it's, this has been a fantastic conversation. So thanks very much for joining me today. The sort of the experiences that you shared, the insights that you also shared, I'm sure are going to be of great interest to our audience. So if after listening to this broadcast, you'd like to tune into future conversations in healthcare, follow our LinkedIn page, because that's where we'll be posting alerts to future episode releases. So in closing, I'd like to thank Carolyn again for joining us, and also thank all our listeners for tuning in. So until next time, stay safe and healthy. I'm Mike Ward, and I'll see you in the next episode.